Hi guys, I'm here with Dane Trembath, who is a reptile specialist here on Grit Island, and as part of Bush Blitz, Dane has agreed to answer your questions. So thanks for joining us, Dane.
Thank you very much, Con.
Dane, the questions come from the Year 7 science class that Mr. Wilkes has put together. And so the first question that the students have asked is, have you found any new species on this trip?
Well, that's always the question people ask us. Possibly yes, but we won't know till we get back and do some more lab work. So basically the main thing we've done on this trip is collect animals for genetic analysis. Most of the new species to find out there have what's called cryptic speciation, whereas they basically, two species might look exactly the same.
Right.
But are highly different. And so what we're looking at is groups of species that live across northern Australia that possibly, Grit being the outlier, could have a different individual, but it could look very much like the same species. So,
right, okay.
Yeah, but I'm pretty sure we've got a new species of gecko on this trip that was known to live on this island, but I'm pretty sure we've collected some.
That's incredible.
On the trip, yeah.
Great. What sort of challenges are there when you're looking for new species?
Okay, basically the main challenge we find is trying to get to remote places. Bush Blitz, of course, helps out with that. Most of our target here is getting into the sandstone. A lot of the lowland around here has been sampled, so having Bush Blitz with this project and being able to get on the helicopter to go out for the night. So I don't have to do a lot of day work. It's actually too hot for reptiles, and other than the pitfall lines we've got going, so most of the time we get dropped out every other night on the scarp. We collect our reptiles and bring them back for processing.
Okay. Where do you put the animals when you find them?
Okay, so basically when I find the animals, they're bagged up. They're incredibly important because there's a lot of money in this trip. Basically, most of our animals are put into plastic bags. They're all labelled. They then go into an esky that sits in camp, and that's like a little treasure trove. And then when the helicopter arrives, they're loaded onto the helicopter. I always insist they go in the cabin. And not outside because they're very valuable, and then basically come down. We then process the animals, and basically what's going to happen with all the animals on this trip is they're going to go to two spots. One is the Museum and Art Gallery of the Northern Territory, and also the Australian Museum where I'm from. We're going to take a subsample of animals to there, but we're collecting on behalf of the Museum and Art Gallery of the Northern Territory. So.
Fantastic. And the next question is, how many years have you studied Grit?
I've studied basically for Groot many years. I've been working on some snakes that occur in Groot Island. We collected a specimen of it the other day, the one that was stuck in the netting.
What animal was that?
That's called a pygmy mulga snake, and that's a snake that we're looking at. It possibly has this cryptic speciation where there could be more than one species. It occurs across the top end of Australia. So basically I have been studying the reptile fauna of the Northern Territory for about 17 years.
Wow.
Yeah. And I lived in Darwin for 14 years, and I've just moved to Sydney to take up the herpetology technician position at the Australian Museum.
Next question is, how do you name a new species?
Okay, well there's probably two parts to these. So the naming comes down to, it's sort of art, So often you might name a new species after someone, usually someone important or famous, or often has had a contribution, either finding the species, a contribution to herpetology, or some tie to the land. The second way you would name them is also based on their morphology, so if they've got long toes or eyelash scales or something interesting. The third thing that's also becoming is using Aboriginal words to name them, as long as you can decipher them, and you will only do that for if the animals are totally indigenous to one area, to one language group, because you've got so many language groups across, you sort of try to, if it's endemic to that area, it's got an Aboriginal language name.
Great.
That's a nice way to name them.
A follow-on question from that is, if you find a new species, can you name it after yourself?
Well, no, you can't. Basically, it's not considered a good idea to do that, but you can name it after your mother, your father, or your brother.
And one of the scientists here, Adam, named it after his dog.
Yes. Yeah. You can name it after pets and family, and basically whatever you want to name it after.
Next question, are there any animals you have found that have been difficult to classify?
100%. There's a couple groups of lizards here that are very hard to identify in the field, mostly because they're so small. The other group that we have difficulty identifying are the blind snakes. They're very small snakes. They quite look alike. The differences are minute.
Yep.
And you have to look at them under a microscope, a low power microscope, to see any difference. So we're just, when we get them, they're always fun to work out.
The next question is, what is the biggest animal that you've discovered?
Discovered on this big bush pit. Probably the biggest animal that I've worked on that's a new species would be some turtles.
Yep.
There's some freshwater turtles in the Northern Territory that are possibly undescribed, and they're in the 3 to 5 kilo range. And once again, it's the same thing. It was a species that occurs across all of Northern Australia, but with the new DNA sequencing, with the genetic evidence, we find that there's actually pockets that are quite different to each other.
Yeah, sure.
Yeah.
The next question is asking, are there any rock paintings that show a strange or different animal?
100%. There's lots of this. Not so much in reptiles, but there's a lot of mammal stuff. There's some quite interesting rock paintings of thylacines and things, but they might actually not be thylacines. So they could actually be dingoes, but no one knows because it's quite hard to date some of these paintings.
Sure.
And some are recent, some are very old. I've seen some very interesting turtle paintings that I find quite fascinating because I'm into turtles.
Yeah, sure.
Yeah, so I've seen one that depicted a species of turtle that I don't think actually occurs in Australia, but the painting shows exactly
fantastic
a species. So might be here.
That's great.
Yeah.
One question more for the senior students, Year 11s. In terms of classification and discovering new species, is the preferred method now DNA, or still we're looking at the physical attributes of the animal?
Well, 100%. So DNA is just a tool, but it's basically become the norm. It's a lot easier to do. It's a lot cheaper to do, but you do need new specimens to get the tissues.
Yep.
Which is why this is great. Basically, you will use your DNA first to look at the groups, and then once you can have strong genetic divergence for those groups, you then can really look at the morphology. But they both really need to work in tandem, because if you can't identify the species in the hand, it's not that useful.
Yep.
Yeah. So because the problem is we name and describe the species and study them, but if people can't ID them in the field, it's kind of not that useful.
Sure.
Yeah.
Understand. Thanks very much, Dane, on behalf of the students of Nhulunbuy High School. Thanks very much for taking the time.
No worries, mate. Thank you very much for coming on the Bush Blitz.
Cheers. Thanks again.
Okay.
Awesome.